Hello everybody, thank you for coming and willkommen zu Freunden und Family in Deutschland. Close enough. Many of you know Chistine Ewers-Saucedo comes to us from an island, Pelvorm, off the coast of Germany. I was introduced to her on another island, Sapelo, with the University of Georgia Marine Institute. We were both studying coastal population biology, both interested in molecular systematics of crustaceans, and I was lucky to get her to come to the University of Georgia. Chistine started working with me on barnacle population genetics, no surprise for those of you who know me. Got 2, soon to be 3, maybe more papers out of that work. That work happened to lead her to another island that ended up being quite important in her life for different ways. But today is not about phylogenetics of box crabs. Today is not about biogeography of Chile. But about Chistine's dissertation on the evolutionary dynamics of an even more intriguing question involving a barnacle, and I'm going to let her tell you about that. But about Chistine is everything as a biologist. She's fieldwork, check, with intensity. Taxonomy, museum work, check, check. Time at the microscope, time with microsatellites, genomes, rearing animals, time just watching them, theory, stats, coding. She's got it. Check. And a genuine pleasure to work with every day. She got her diploma in biology at Christian Albrechts University in Kiel, came to us via the old ILS program, and has signed on for a postdoc at UC Davis with Rick Grossberg, my old boss. So I'm really pleased to present Chistine Ewers-Saucedo.
Well, thank you, John, for your kind introduction, and thanks to all of you for supporting me during my dissertation defense today. My dissertation aimed to explain a rare and unusual evolutionary phenomenon, the coexistence of males and hermaphrodites. Hold on, let me begin. My dissertation aimed to explain a rare and unusual evolutionary phenomenon, the coexistence of males and hermaphrodites. And I am there. This sexual system, called androdioecy, has fascinated and bewildered many evolutionary biologists before me, one of them being Darwin. Darwin wrote, "It is so novel a fact that there should exist in the animal kingdom hermaphrodites, aided in their sexual function by independent and, as I have called them, complementing males, that a brief consideration of the evidence will not be useless." So after the discovery of androdioecy in barnacles, Darwin felt it necessary to explain his evidence in detail, because he knew if he would fail to do so, his peers would not believe him. This disbelief has never ceased to exist. I think it is mainly because the conditions needed for androdioecy to evolve are so unlikely to be met in nature. Even Darwin, otherwise extraordinarily proficient in explaining evolutionary processes, did not have an answer for this phenomenon. He wrote, "Regarding the final cause of the much more singular fact of the existence of the complemental males, I can throw no light." So please let me try and explain why androdioecy should not exist in nature. Androdioecious populations consist of hermaphrodites and males. But while hermaphrodites can reproduce through both the male and the female function, here we have eggs and sperm to represent this, males can only reproduce through the male function. So you may ask, why be a male when you could have it all? Think of it as a cookie baking competition, where hermaphrodites have a big KitchenAid to make cookies, while males only have a small whisk. Do you think males could ever bake as many cookies as hermaphrodites? In other words, how can males ever compete with hermaphrodites? And the resource these two sexes or phenotypes are competing for is fitness. It's the amount of genetic material passed on from generation to generation, or it's the number of genomes that are passed on. And what we can see here is that hermaphrodites, because they're the only source of eggs, already move half of the genetic material, have already half of the fitness, because each offspring has 2 genomes, one from mom and one from dad. As you know, we have half of our genes or gene variants from mom and half from dad. So they already have half of the fitness. So the remaining half of the fitness has to be separated or split up between males and hermaphrodites. They kind of compete for fertilizing offspring. And in order for males to even occur at the lowest frequencies, they have to fertilize twice as many eggs as hermaphrodites. But in a world where sperm is cheap to produce and overabundant, this twofold fertilization success appears unlikely to occur. And so given this unlikeliness, many biologists have often questioned whether androdioecy can in fact be an evolutionarily stable state, whether males can be maintained with hermaphrodites because of their fitness. Or if conversely androdioecy is just a transitionary state between hermaphroditism and dioecy, the two more common sexual systems. And then secondly, what are the conditions that would favor androdioecy? Despite the unlikeliness of androdioecy, we think we see it confirmed about 160 species nowadays, both plant and animal species. And the detailed study of these systems has revealed some astonishing similarities. Hermaphrodites always appear to be sperm limited and use the male function predominantly to self-fertilize. Males have some sort of mating advantage. May that be that they actually produce 6 to 10 times more pollen or sperm, or that they are the preferred mating partners. And lastly, all of these systems come from dioecious ancestors, so from separate sexes. And so Pannell developed a model that seems to fit these observations quite nicely. He proposes that androdioecy can evolve from dioecious ancestors in populations or in species that frequently colonize new habitat. So what we see here is that we have an ancestrally dioecious population, and as it colonizes new habitat, we can assume that in these new patches of habitat, the population densities are very low, and the number of mates that these individuals have is also very low. So it would be highly advantageous for these females to gain the male function in order to be able to self-fertilize their own eggs. And then as populations grow, males can now invade these systems, and we have androdioecy. But you can also easily see that these systems could revert back to the dioecious state. So under this model, is androdioecy evolutionarily stable? I would say not really evolutionarily stable, but it can be maintained over evolutionary times as long as this colonization cycle is kept up. And the conditions under which androdioecy evolves are when new habitat is frequently colonized. So this is it, right? The end, I'm done. Well, I wouldn't be standing here in front of you if that would be the case. And if you remember back to the very first slides, there is a taxon that has received much less attention than the other species I just put up here, and that is barnacles. So the aim of my dissertation was to investigate the evolution of and maintenance of androdioecy in barnacles. Barnacles are well suited to study androdioecy because it evolves repeatedly from hermaphroditic ancestors. And this is where it differs from the species I talked about previously. And so here's a partial phylogeny from Ewers et al., 2012, and we see at least 5 instances of androdioecy evolving from hermaphroditic ancestors. Moreover, barnacle males always have a peculiar phenotype. They're always dwarfed, and they're always attached to larger hermaphrodites. From this figure, you may or may not be able to see that there are also dioecious barnacles. So in this figure, we have these big, funny-looking things that are the females or the hermaphrodites, and then these small, funny-looking things, which are the males. So I think the question still persists whether androdioecy, even in barnacles, is only a transitionary state between dioecy and hermaphroditism. And so based on what we know from barnacles and based on what we know from androdioecy, I proposed 3 hypotheses that I was going to test. The first hypothesis is that in barnacles, the males have a mating advantage. Males have a mating advantage because they're really close to a mate. The hermaphrodite they are attached to. and proximity increases mating success. This advantage is further accelerated when hermaphrodites are sperm limited. Secondly, or in contrast to that, we could imagine that androdioecy is maintained by differences in life history. Male dwarfism is often a sign of early maturation and higher survival to maturity. If males would have a relatively higher survival to maturity, that would increase their relative fitness relative to hermaphrodites. And then if this is the case, androdioecy should have evolved in populations with high mortality rates or low growth rates, because those are the conditions where early maturation is particularly advantageous. I aim to test these hypotheses in the epizoic barnacle Chilonibia testudinaria. And Chilonibia testudinaria occurs worldwide, and also here along the east coast of the United States, where my studies took place. And so it occurs on a number of animals, but I was able to collect them from loggerhead sea turtles, horseshoe crabs, and blue crabs. And I was able to collect a quite large number of them. I was able to collect almost 5,000 barnacle individuals from more than 500 host individuals. And the following studies that I'll be talking about draw their samples from this large collection. I just realized that that also means I killed about 5,000 barnacles, but that's a biologist. So when we look at these barnacle communities in situ, so this is on the back of a loggerhead sea turtle on board of the RV Bulldog, where I did a lot of my collections. What we see here is the hermaphrodites, which are both these big pink barnacles, as well as these smaller white barnacles attached to the host itself. And then we see these males, which are the barnacles attached to the hermaphrodites. And so this is the way I and some others define the sexes just by their attachment location. All right. So now I'll begin the data-driven part of my dissertation, and I will begin by talking about the first hypothesis, whether androdioecy is maintained or facilitated by the mating system. And if that would be the case, then males would have a mating advantage, which would likely be caused by their proximity to the hermaphrodite they are attached to, so they should fertilize most of the eggs of that hermaphrodite, and by the sperm limitation of hermaphrodites. Barnacles are great for studies of mating systems because they allow us to collect complete mating assemblages, or they allow us to collect They allow us to collect mom and dads or all possible mates. So what we see here is a horseshoe crab that I collected, and all the individuals on it, I think, can mate with each other. So that's what I call like a potential mating group. And in order to understand who actually did mate with whom, I used parentage assignment. So barnacles, well, let me step back. Barnacles also, in order to copulate or to mate, have to use really long penises, the longest penises in the animal kingdom in relation to their body size. So we know the mates have to be all on this host. And then barnacles also brood the offspring, so the eggs are fertilized, and then the hermaphrodite that acted as a female will brood the offspring for, we think, 1 or 2 weeks. So if I collect brooding hermaphrodites, I can assign the paternity of these embryos that the hermaphrodite is brooding. And from that, I know that those are the mates of at least one mating event. And that is exactly what we did. And we used microsatellite markers that I developed in the lab and genotyped potential mating groups at about 3 loci. If need be, so if we couldn't assign dads unambiguously, then we just used more microsatellite markers. We had 12 total. Then we assigned the parentage via the exclusion method, and we determined that all dads are the mates. And from now on, I will talk about the mates, but basically they're the dads of the brooding embryos. One last point. We said that only the males that are attached to the focal hermaphrodite, so the hermaphrodite that we genotype the eggs from, would be a potential mate for that hermaphrodite. That has been shown in literature several times. So we adhere to that too. We were able to determine the mates of 54 potential mating groups, which means we were able to determine who mated with 54 different focal hermaphrodites. And we genotyped more than 600 adult individuals and 800 embryos. And so the results were actually, in the beginning, were very surprising to us. So what we see here on the left-hand side is all the potential mates. So all these more than 200 individuals we genotyped. And we see the distribution of hermaphrodites and males. And then on the right-hand side, we see who actually fertilized or who actually mated with that focal hermaphrodite. And the only thing I want you to notice is that these proportions are almost identical. If males had a mating advantage, we might have expected this pie chart piece to be much larger. We also wanted to look at this in more detail. So we asked what determines mating success using distance to the mate, penis length, and sex as explanatory variables. And then we used conditional logistic regression to assess the significance. And these results, again, surprised us. So what we found is that actually being closer to a mate made it more likely to be a successful mate. So here we have a histogram, a distribution of the distance of all potential mates here in gray, and of the actual mates here in the shaded bars. And what you can see is that the actual mates are on average closer, than the random or than the distribution of potential mates. We also found that penis length was important, so having a longer penis made you more likely to be a mate. This doesn't show us nicely on this graph, probably in part also because it's not as significant as the distance, as the proximity. And this, I'm sorry that this cut off somewhat, but basically what it just says is that being male, even in this analysis, never showed up as being advantageous for mating success. So being male did not increase mating success. So how can we make sense of that? Well, when we looked at the sex-specific differences in both distance and penis length, we found that, not surprisingly, males were always closer to the hermaphrodites, to the focal hermaphrodite, because they are attached to it. But we also found that they had a shorter penis. So one possible explanation is that these two factors kind of offset each other, and in the end lead to no male mating advantage. And we also see that hermaphrodites seem to be doing just fine in mating. They do not appear to be sperm limited. So what we can conclude is that the mating system does not seem to be responsible for the evolution of androdioeci in Testudinaria. So if not the mating system, what could be responsible? And this is where our second hypothesis comes into play. Well, first, do we see sex-specific life history traits? And secondly, if so, do these traits provide an explanation for androdioeci? And this is because we know each host represents a very different habitat for the barnacle. We did all these analyses on a host-specific basis. We do not see genetic host specificity. I tested that using the microsatellites I talked about earlier, and other people have looked at that with both mitochondrial and nuclear sequence markers. So we don't think there is any, as far as we can tell, there's no genetic host specificity, but we do know there's a lot of plasticity and other traits as well on each host. And so we hypothesized that given the small size of males mature at a younger age. Consequently, males have a higher relative survival to maturity. But throughout the following part, also talk like a higher viability. And this would increase the male fitness relative to the fitness of hermaphrodites. And in particular, we can test the evolutionary significance of these life history traits by fitting empirical values to models of androdioeci. And so we looked at two different models that could help us understand the system. And the first one is kind of a minimal requirement for the invasion of males. And that is that the relative viability which is the ratio of the hermaphroditic viability divided by the male viability, should be larger than a half. And the way you can think about it is that males should be at least twice as successful in reaching maturity than hermaphrodites. And if this is true, then males can invade the system. Secondly, we fitted the model of Lloyd, which we were able to simplify. So the original model of Lloyd had some mating system parameters in there, which we now have shown that mating system does not play a role. So we were able to simplify the model to only include, again, this relative viability. And yes, and the response, or what we can do with this model, why it is nice, it gives us the expected or the predicted male frequency of the system. So we can actually compare that to empirical data as well. So as you have seen, the relative viability seems to be the important thing that we need to calculate here. And when we basically calculate out what that means, it is the ratio of the survival, it is the relative survival multiplied by the relative age of maturity. And it is the survival of hermaphrodites divided by the survival of males, multiplied by the male to hermaphrodite age of maturity. And you can think about that, because I know math is always tricky to look at, but you can think about it, that hermaphrodites should do better the higher their survival rate are, the more should survive to maturity, and the faster they mature, right? So the smaller this is and the larger this is, the better hermaphrodites should do, and vice versa for males. And so first, let me explain how we looked at this relative survival rate. Okay, that's fine. So we follow Charnoff in that we say the biggest differences in survival rate between the two sexes occur when the larvae settle. So I've told you that barnacles brood the offspring for 1 or 2 weeks, but then they release the larvae into the water column, where they spend another 1 or 2 weeks, and then they have to find a place to settle, where they then stay for the rest of their life. So for hermaphrodites, when they are ready to settle, they can settle on any host they have available, right? Or that's at least what we assume. Whereas male larvae can only settle on a host that already has hermaphrodites and large hermaphrodites on them. So they always have a lower survival rate. And in this case, male larvae would have like a 1/3 survival rate for horseshoe crabs, and the survival rate would be a half of that for when they try to settle on blue crabs. And because we're only interested in the relative nature of this measurement, we can actually set the hermaphroditic survival rate to 1, and then the male survival rate simply becomes the proportion of hosts with hermaphrodites. And so we estimated this proportion during our field surveys, which spanned 5 years, as well as from literature values. So lastly, or the next part we needed to look at was the age and maturity. And we determined the age at maturity by basically the penis presence. So first we looked at the presence of the penis, and we used it as an indicator of maturity. So obviously they need a penis to be able to copulate, to be able to reproduce. And so we assessed penis presence in males and hermaphrodites of different age classes, and then again we used logistic regression to determine when about 50% of the population were mature. And secondly, we also used penis length, and here the age at maturity was when penis length was 0, so when it intercepted with the age. And we used this, and we basically assessed this, this point with linear regression. And again, we measured penis length of males and hermaphrodites of different age classes, and then with linear regression against age, found that point where penis length was 0. Lastly, we had to calculate some male frequencies, because they are the they are what is predicted in the model of Lloyd, and we did that simply by counting the number of mature individuals per host individual and then calculating male frequencies per host species. And so here are some of the results that we have. I think some of that might be hard to see, but I'll walk you through. So on the left-hand side, we have the results of the logistic regression. So where we just looked at penis absence or presence. And so when there's a 1, this means all individuals have a penis, and a 0 do not have a penis. And so this is the probability of penis presence. And what you can see here, so these are little m's, which stand for males, and these are H, which stand for hermaphrodites. What you can see is that males mature at a younger age than hermaphrodites. And the colors denote the 3 host species. So from this, you can also see that it appears that loggerhead sea turtles, or barnacles on loggerhead sea turtles, always mature earlier. On the other side, we have the results of the linear regression, where we have regressed penis lengths against age. And as you may note, this is not the biologically correct way of plotting this, right? Because this suggests that penis length determines age. I am aware of that. But what that lets us do is calculate really nice error bars around this estimate. And so this is here where we are interested in, where penis length is 0. And I know this is kind of hard to see. So I also wrote it out in a table. And what we can see here pretty clearly is that hermaphrodites mature after 4 to 6 months. The results of the two, like the penis presence and the penis length estimates, were pretty similar. So I only present here the results of the linear regression, while males mature only after only 1 month. So males mature at a much younger age. When we then looked at the male survival, so if you remember here, we're now at this part. When we looked at the male survival, what we saw is that the males should survive the best when they are trying to settle on loggerhead sea turtles, and that makes sense because almost every loggerhead sea turtle has large hermaphrodites of testosteronaria on them. When we now calculate this relative viability, this value we are so interested in, we see that it is only smaller than 0.5 or a half on loggerhead sea turtles. So only these hosts seem to provide the minimal requirement for androdioecy. In concordance with that, if we use Lloyd's model and calculate the frequency of males, we see that there should not be any males on blue crabs and horseshoe crabs, but a high proportion of males on loggerhead sea turtles. However, if we compare that with empirical values, we find that these values are all quite similar, and that they're all between, let's say, between 0.3 and 0.4 up to 0.5 maybe. So how do we make sense of that, right? So we have shown that life history traits are host-specific, but without genetic underpinnings. I talked about that. But assuming that sex determination is genetic, the observed male frequencies may represent some kind of fitness compromise, or they and that's the best way I can explain this. Like they should be really high on one host, and they should be 0 on all other hosts, but they kind of they wash out, right? And we may be able to assess if that is in case the fact if we estimate relative barnacle abundance. So if we are estimating the number of barnacles on total on all of blue crabs, all the horseshoe crabs, and all the loggerheads, and then calculate the relative values, we see that most barnacles occur on blue crabs and on loggerhead sea turtles. And then if we multiply basically 0.42 with the high male frequency that we saw based on Lloyd's model, we get to a value of 0.3, which is much closer to what we actually see in nature of 0.3 to 0.4. And so we think that provides some evidence for the idea that survival to maturity for males on loggerhead sea turtles is high enough to maintain androdioecy in the system at large. So onto our last hypothesis. So we have now shown that indeed there is that there are 6 sex-specific life history traits, and they do seem to provide an explanation for androdioecy. So do we also see either high mortality rates and or low growth rates that would facilitate or that would be very advantageous for early maturation? And so in order to get that, we had to estimate growth rates. And I did that by basically every approach I could think of. We did lab trials. So this is what you see at the bottom left here. This is the carapace of a blue crab. So it's a dead blue crab, but it still had a barnacle on it. And when you look closely, you may be able to see that there's like this white rim at the bottom of the barnacle. And that's the amount the barnacle grew in the lab, because the water and the conditions are just cleaner. We used mesocosm trials, and this is what you see here on the far right. So this is a minnow trap, a small trap with a blue crab with barnacles on it, and we buried these in the intertidal, and basically to try to get a little bit more natural estimate of growth rates. We used field observations, and that's something that fish people may know more about. We basically computed size frequency spectra, so we assumed. So here, this is what we did. We looked at all these barnacles on the host, and we saw that there were 2 size classes. And we assumed that the large barnacles are 1 year old, and that the small barnacles, so this is all these white dots, are basically really small, recently settled barnacles, are less than a month old, are recently settled. And then we are able to use the difference in size and assume that that's how much they grew in 1 year. And then we also had some recapture pictures from loggerhead sea turtles. So people, are really interested in loggerhead sea turtles, and they will go out and they will catch them and look at them and take pictures. And sometimes they just recapture them, 3 months later. And we were able to identify certain barnacles on these loggerhead sea turtles and determine how much they grew in that time. To look at mortality rate, we applied these growth estimates to our field collections. So all barnacles that we collected, we measured the size, and so we were able to estimate the age for all of these barnacles. And then the mortality rate, we used a rather simple approach, which is inversely related to maximum age. And intuitively, that makes sense. The higher the mortality rates, the younger individuals die on average, or the lower the maximum age should be. And then because we were interested if the values that we got for Calanivia testudinaria were unusual, we compared it to literature values of other barnacle species. And we only found purely hermaphroditic barnacle species that these kind of values have been measured for. So all the values you see are just from purely hermaphroditic barnacles, without males, that is. So just one slide to the results here. What you see is that it all kind of looks the same, right? So what you see here is everything we did for blue crabs, for horseshoe crabs, and for loggerheads. And here at the bottom, you see basically what kind of approach we took. So these are the laboratory trials, mesocosm. These are the size frequency spectra, the field measurements, and so on. And that you actually don't see a whole lot of a difference is good, right? It gives us more confidence in our estimates. However, when we actually did the statistics, we see small differences, and taking out the least natural conditions, we were able to show that these estimates are indeed host-specific. When we looked at the age structure, we found that hermaphrodites, the maximum age for hermaphrodites was 600 to 800 days, so maybe 2 years maximum, whereas the maximum age for males was less than a year, so they died much younger. And that is not surprising if you remember that they settle on older hermaphrodites. So the moment the hermaphrodite dies, they die as well. And so when we compared our results to the literature, we found that the growth rates for Calanivia testudinaria were somewhere smack in the middle of growth rates that had been estimated previously. And one thing that I will say is that we had only very few estimates for males, but they were somewhere in the range of the hermaphroditic growth rate estimates, so we simply assumed that the growth rates are the same. But I think more work could be done on that part. But however, what you do see is that the mortality rates, both for hermaphrodites and males, are much higher than the mortality rates for most other species. And the only two that are somewhere, or that are within the range of Calanibia testudinearia, are two small thamelids, which are the barnacles that John and I work on as well, which are high intertidal, really small barnacles. So we think that provides some evidence that Calanibia testudinearia has indeed a higher mortality rate than most barnacles, but the growth rates are similar. But these higher mortality rates are what we expected to see. So zooming out a little bit, so do we think that androdioecy is evolutionarily stable in testudinearia? We do actually think that might be the case. But in contrast to other androdioecious systems, we think that it is actually due to sex-specific life history traits and not at all to the mating system characteristics. And then the conditions are conditions where mortality rates are high. And if you think about it, living on the back of a loggerhead sea turtle or a horseshoe crab, there is high mortality because the sea turtles shed their carapace all the time. Horseshoe crabs as well, or they die early, and blue crabs have a high mortality rate as well. So these are kind of unstable habitats. And so that is the one point where I think they all kind of are similar. So despite androdioecy being maintained through very different mechanisms, all these species seem to occur in somewhat unstable environments. So we have this little crustacean that I showed in the very beginning. This is a freshwater crustacean who lives in basically big puddles, temporal ponds. I had a plant up there, the Tisca glomerata, which occurs in wetlands in California, which by now must be pretty rare. And then the last one was C. elegans, and as far as I know, it occurs, it's kind of like a soil nematode, which also faces frequent extinctions, local extinctions. And so Calanibia testudinearia has kind of the same unstable habitat. All right, and so with that, all right, I would like to thank so many people. I would like to thank my family and friends in Germany, without whom I would have never made it through. They never made me feel bad for leaving them so far behind. And have just been such tremendous support. I would like to thank my friends and family here in Athens, again, without whom I would not have made it through this. Special thanks to Neva, who basically single-handedly generated all the data for the first part that I showed you, the mating system study, and our newest family member, Maximilian, who is sleeping. Everybody who knows me a little bit knows that I'm kind of crazy about soccer, but I think that actually helped me keep my sanity and was a good other challenge. When I started grad school, I did not play soccer. Now I score about 1 goal a week. I'm very proud of this. I had to point this out. And so lastly, I would like to thank all of you. I would like to thank Athens for giving me such a fabulous time here. And, I could not have made this. I would have never started this without you, Daniel. So thanks for hanging on. And with that, I'm done with my talk. I'm open for any questions. If there's still the question, why work on barnacles? This is me doing field work. All right, thank you. All right. Yes, Dave.
So I think I misunderstood one of your data slides, which was, did you say that basically half of the barnacles population exists on blue crabs? Or did you say that blue crabs, about half of blue crabs have barnacles on them?
No, half of the barnacle population occurs on blue crabs.
And like 30-something% on sea turtles?
Yes, and half of them. So, and
is that surprising? I mean, I would have guessed like 95% would have been on blue crabs, because I would have thought blue crabs is a much, I mean, like
Much more
substantially-
abundant. So you're right, and so there are two different things happening. So on blue crabs, about on average 10% of blue crabs, only 10% have Calanibia on them, while most of loggerhead sea turtles have Calanibia on them. In addition, we see, let's see, what are the numbers? We see on average maybe 5 or 6 Calanibia on a single blue crab, while we see maybe anywhere between 300 and 500 Calanibia on a sea turtle, on a loggerhead sea turtle. So I think there where,
And so, but how many turtles are there per blue crab out there in the world? I mean,
There
I
is
would've
an
guessed like 10,000,
astonishing-
but maybe I'm way off.
there are more, way more. I can't remember the number. There is an astonishing number of loggerhead sea turtles out there. So I go out, the one slide I showed where I was on board of the RV Bulldog. So they go out and their sole purpose is to catch loggerhead sea turtles. And they have like it's a shrimp boat, right? And so on any given day, they will catch anywhere between 10 and 20 loggerhead sea turtles. That's short trawls, 10 trawls a day. So I was surprised too. There is a high number. But these estimates are indeed very fickle because really we still don't really know how many blue crabs there are, the horseshoe crab estimate is the worst. We really don't know how many of those there are. And even, for loggerhead sea turtles, we really don't know, which is why this research is still undertaken. So this was kind of my best guess. But you're totally right. If those estimates change a lot, then that could change the overall outcome. But we just don't know. Yeah, Andrea.
Excuse me. So just going back to the beginning of your talk, when you said that there are about something like 150 androdioecious species out there, and that in all other cases they evolved from dioecy and these other things. So surely that's not known for all 150.
So what is?
Like how many do they really have good phylogeny?
So what is happening is that in animals, when it occurs a lot. So in these nematodes, so C. elegans is the one most of us know because it's a genetic model organism. But I think there are another, like 20 species in that same kind of clade that are all supposedly androdioecious. And then these small freshwater crustaceans, there are another 20 in that same kind of clade that are androdioecious. So I think there are still different things happening between plants and animals, because what we see in plants more is there's like one terminal branch and androdioecia occurs once, and then you don't see it in the whole genus or family or whatever anymore. Whereas in animals, and then for barnacles on itself, I think there are about 60 barnacle species that are supposed to be androdioecious. And so this estimate of the 150 actually included the barnacles. I should have said that. So that's just all species overall. So I think that's where we can see. And you're right, we don't, there are certainly still cases where on the one hand, we think they're androdioecious, but not many studies have actually done paternity testing on them to actually show that or even like mating trials to show that all of these species are androdioecious. So the number could be lower. But those are pretty confirmed cases. And in most cases, it actually do come from dioecy.
So in these barnacle cases, is this like one evolutionary event? And then, so is it phylogenetically that you have hermaphroditism paired, like happening 20 times? Is there 20 events?
It's not.
So is it like a one evolutionary event and then 20?
So in that phylogeny I showed, we saw at least 5 independent events. What we do see, so what I can tell you for Calanibia testudinaria, I said it occurs worldwide, but really there are probably 3 different species, one species in each ocean, but they are all, as far as we know, they are all androdioecious. So really there would be then 3 species that are androdioecious. And so we do see some of that where it's one event and then it just spread out. We do seem to see like events where it's like androdioecious, dioecious, hermaphroditic, and I think there's still more work to be done to really like suss out the phylogeny, but people are on that. So I think we see both, and then the sister species at large of Chelonibia, outside of these 3 closely related species, is purely hermaphroditic. Yeah.
Can you tell from your microsatellite studies whether the hermaphrodites are actually fathered by males or from selfing?
I have never seen a case of selfing, and we have looked. So a part that I haven't talked about is we were actually really interested whether they self-fertilize. So for all the part that I looked at where I determined the mates, right? We always found a paternal genotype in these embryos, but then we also specifically went out and collected isolated hermaphrodites, right? With these hosts, it's pretty easy. Sometimes you'll only see one large hermaphrodite on a host. I collected these. There were, out of 120 or so that I collected, there were actually 2 that were brooding. So I was like, oh, self-fertilization. But everybody else was not brooding. But when I genotyped those offspring, they still had a paternal genotype. So either there was another barnacle around and just fell off, or maybe the hosts interact really closely. So not a single case of self-fertilization. And we really tried. I think that would have been really cool, but not in barnacles, yes.
So with these androdioecious species, were the ones that they can figure out, is it clear that they're genetically determined, or are there facultative?
Yes, the sex determination. That's actually a good question. So in most cases, it seems to be genetic sex determination. In some cases, it is both genetic and environmental, so there's some wiggling space. And for barnacles, the few cases that people have looked at, it seems to be the same way. Some species appear completely under genetic control, the sexes, and in some cases, it's a little bit of both. And yes, you're right. That would be definitely something I would be really interested in doing in the future, looking at that in Chelonibia testudinaria, because we do not know that.
In cases where it's environmental, does the advantage fall apart? Is it in those situations, is it sort of making best of a bad lot or low resource allocation that produces the patterns, or does it still fit into this framework?
Well, it depends. Let me think about this. So within the barnacles, where it is both, what is happening, and we have exactly 2 cases, that's why. We have one case where it's completely genetic, and we have one case where it's something of both. So all individuals can be hermaphrodites, and about 50% can also become males if they find a hermaphrodite, right? And I think in barnacles, a lot of it depends on whether there is a hermaphrodite available to settle on or not. And in plants, as far as I can recall, it's more about resource availability in the cases that I recall. All right. Thank you so much for listening in.